All right, well, I'm going to reprise a talk that Bob Orth, John Lefchak, and I, Bill Dennison, gave to the Water Quality Goal and Implementation Team in November. And the talk is about the SAV, Submerged Aquatic Vegetation Status and Trends. We had a group, we called ourselves the SAV-SYN, the SAV Synthesis Group. And we did this because of timeliness, in several manifestations of timeliness. One was that we had long-term solid data sets available. We had access to some new analytical tools and expertise. We needed to understand the drivers of the SAV trends because of the important management relevance that those drivers have, and timely input to the 2017 TMDL reassessment. We also, Bob Orth and I, in putting this group of scientists together, wanted to transition to a new generation of young scientists. This is something Bob and I have been envisioning for nearly a decade. We put a proposal to the Bay program back in 2009, and we talked about the estuarine science phase, the eutrophication phase of the Bay program, the coordination of the modeling and the monitoring, and then this accountability phase, which led us into the TMDL. And we envisioned a future in which we use good synthesis. So this opportunity that presented itself with the SAV trends was a fantastic opportunity that we saw to do a synthesis in light of the Bay program in a management context. So one of the things that we've hypothesized from looking at the report card that we've done for the last 11 years is that there are these tipping points that occur in the Bay, and some of these tipping points have to do with the resurgence of SAV. And when we get aquatic grasses, the baffling and filtering that those grasses do removes nutrients and suspended sediments, binds and traps the sediments and creates internal water quality, and that those create positive feedbacks and result in improving health, and those are particularly manifested in the Upper Bay. We also have the premise that SAV are coastal canaries. They're the things that act as a very sensitive, long-term integrated indicator of water clarity and water quality overall. They integrate environmental conditions, they're responsive to perturbations. The fact that we have SAV throughout the Bay helps, and they're ecologically important as well. So like this miner that was looking at a canary, we replaced the canary here with the SAV. And remembering what happened when the canary fell over in the mine, that gave the humans enough time to grab their cage, take them out, and save themselves and the canaries. And so the SAV represents our canary in Chesapeake Bay. So Bob and I were very careful to come up with a small group of excellent scientists to focus on analysis and interpretation that have a commitment to Chesapeake Bay and a willingness to work collaboratively toward a common cause. And we emphasized the word praxis, thoughtful, practical doing in this work group. So we wanted thoughtful people, but it had to be practical and we had to get it done. So here's the team of diverse and talented scientists. Bob and I over here, generally, there are a few older folks here like Don Weller and Ken Moore, but Jenny Kiesman, Cassie Gerbitz, Chris Patrick, Brooke Landry, this is Rebecca Murphy, Jeremy Testa, and then two postdocs associated with CERC, Mike Hammond, and of course we have Rich Patuk here, who didn't participate directly, but had a really important critical role in identifying the important issues for us to tackle. Now the goals of these synthesis workshops were fivefold. One, we had productive workshops. We wanted to produce something at each workshop, bookmark the event, document the progress. We wanted interactive workshops. We wanted a condensed effort so that we did it all packed in an action-packed couple days. We had participatory, so everybody had input, and we had some fun in the process. So this really was an immersive environment. Even when we were at the Ram's Head after a long day, we were using the beer napkins to come up with a word cloud on key SAV attributes. So we're going to talk about this in four segments. The three papers that we have produced and the segment analysis that's currently underway. And I'll go through these four things in turn. So number one was the eelgrass paper that came out earlier this year in Climate Change Biology. Multiple stressors threaten the imperiled coastal foundation species eelgrass in Chesapeake Bay. And this effort showed with historical maps and distribution assessments that the eelgrass used to stretch all the way up here to Kent Island, and now it's shrunk back to just the lower bay, and none of the western tribs anymore have it beyond just the mouth of the tribs. So it's very reduced in distribution. It's also total cover has gone down, and then we've got two appreciable dips here associated with a 2005 and a 2010 thermal events that knocked the eelgrass back. Eelgrass is near its southern distributional limit, so very sensitive to these hot temperature events. The total cover is particularly diminished in the deep water beds, more than the shallow water beds. So the depth has shrinking. They're getting squeezed into a shallow zone and closer to shore in shallower water. We also looked at, with some models and some data that shows that the declining water clarity combined with increasing temperatures provide a synergistic effect, so that the combination of warmer water and cloudier water results in this eelgrass decline. We did some back of the envelope calculations associated with ecosystem impacts. These are literature values about just a few of the ecosystem services these plants provide: nutrient cycling and secondary production. And we found with the loss rates came up with economic numbers on the order of $1.5 billion to $2.5 billion, depending on the time period that you integrate that. So, the number's a little rubbery, but the significance is not is unmistakable. These plants decline have significant economic as well as ecological impacts. The next paper was a bioscience paper that came out in summer of this year, Submerged Aquatic Vegetation in Chesapeake Bay, Sentinel Species in a Changing World. We were able to get the cover photo of this prestigious journal, which we were proud of. We had a bunch of different graphics. I'll show you a series of 6 photos we used to depict some of the key issues: the chicken expansion of the chicken industry, the hardening of the shorelines, the expanding clam aquaculture, the introduced species like hydrilla, which in the Upper Potomac, some of the restoration of seagrass effort, and then some of the epiphytes associated with nutrient pollution. And we looked at did some modeling on the water quality and showed the hysteresis of as water quality improves, you have recovery, but the crash takes place on a different trajectory. And we talk about the sensitivity and engineering ability. The beds themselves engineer the water quality around it, so they can influence, and the larger the bed, the more engineering takes place. And so that reduces the sensitivity of those beds. We looked at this in a whole bunch of different segments, and this is in 15 different reporting regions, as well as 4 different salinity regimes. And we see the dramatic resurgence in the tidal fresh oligohaline beds, not so much in the mesohaline and the polyhaline dominated by eelgrass, with the heat events showing a general decline. We connected this to some of the overall report cards and water quality, other water quality indicators that relate this to the seagrass. And so we see some of these upper bay improvements here are related in particular to the engineering of those SAV beds that are resurging up there. We also documented the fact that we've got lots of management actions that have taken place associated with grasses. Cumulative management actions have stepped up since late 70s, as well as the number of scientific publications. And so the total number is given here, and that took off in the late 70s. But it's comprised of papers on the animals associated with SAV beds, the distribution abundance of SAV, the processing and modeling, the reproduction and restoration. We also looked at the number of media articles in the popular press, and that has really taken off in the last decade. It's in 2004. And the number of restoration projects, lots of restoration, not as much in terms of the success of those restoration. Some of that's time course, some of it is that you really have to have the water quality to be able to restore these beds. We used some conceptual diagrams in the bioscience paper. We talked about 6 vignettes here from around the bay, the upgrades of sewage treatment, and particularly in the upper Patuxent and upper Potomac that have created resurgences, the resilience, particularly of the Susquehanna Flats, associated with response to Tropical Storm Lee. We look here at the invasive hydrilla of the upper Potomac, and then being able to create suitable conditions for native species recolonization. So invasives aren't necessarily always a bad thing on the long term. We have our boom-bust cycle of our widgeon grass, or rupia, here on the eastern shore, with high runoff events versus low runoff events, and having massive changes from year to year. We have our thermal stress story and our shallowing of the eelgrass beds that we alluded to earlier, and our clam aquaculture. So that's our bioscience paper. Now we have a paper that we're in final revisions with that looks like it's going to be accepted. I guess it really has been accepted pending these revisions to the Proceedings of the National Academy of Science. This one's called Nutrient Reductions Lead to Unprecedented Recovery of a Temperate Coastal Ecosystem. The short title, the TMDL works. So we asked 2 questions. What are the long-term SAV trends, and how are the trends related to human activities? And we tackled that from the watershed and the water column perspective. We used 3 different long-term data sets in this analysis. We used the SAV mapping that's been going on since the mid-'80s, the Chesapeake Bay watershed model, and the water quality monitoring from around the bay. Again, another 30-plus year data set. We looked at sub-estuaries, and we picked these color codings respond to the major salinity regimes, the polyhaline high salinity red, the mesohaline green, and then the oligotrophic to oligohaline to tidal fresh in the blue. And these are sub-watersheds for which then we could relate specific SAV beds to. And then we also looked at the water quality across the entire segment schemes of the bay. And we used a technique called structural equation modeling. This is John Lefchek's, who was a postdoc on this project from VIMS, who's now up in Maine at Bigelow Lab. He did this structural equation modeling, which takes variables in a causal network, and it's great to test cascading effects, and it's a bunch of linear regressions. So you start with 2 variables and say X influences Y, so we can do a linear regression there, and then Y influences Z, and so we can get that relationship, and then we connect it together into a single causal model, a structural equation model. So you start with all these boxes and arrows, and then you work through using the data, interrogating it with the data to come up with the things that are significantly linearly correlated. So in these cases, these are the arrows that showed significant correlation. So the percent of agricultural land in these watershed units correlated positively with fertilizer manure. More ag, more fertilizer, more manure, more non-point source nitrogen. The more developed areas, the more point source nitrogen we had. Then the negative correlations coming together as red arrows, where the total suspended solids plus the diffuse sources, which are combined with flow, and the point sources all negatively influence the SAV. Species richness turned out to be a positive influence, and the amount of available habitat another positive influence. And then with the watershed structural equation modeling, we were able to look at the agriculture to fertilizer to non-point source affecting SAV and the species richness positively. So these are all the things that then started to put together some very strong correlations. I mean, these are R-squared values of 0.8, 0.7, and almost 0.7 in the different regions. So those are very robust across the entire bay. And then we did a water column structural equation modeling. And this is taking just the water quality data from within the bay, not the watershed data. And we use nitrogen, chlorophyll, Secchi that influence SAV, species richness, of the SAV that influences it. And we had both direct and indirect effects of nitrogen directly on the SAV as well as through the cascade. And that R squared was very high as well. So this provided us some really strong, for the first time, really strong direct evidence of some of the impact of nutrient declines that we were seeing across the bay and leading to resurgence. So just kind of stepping back, taking our mean nutrient input, and we have a 24% reduction, but we also concurrently have a 245% resurgence of grasses. So that's pretty powerful testament for the importance of maintaining our nutrient diet, our TMDL, because we're going to get these kind of ecological consequences. So, addressing these questions, what are the long-term trends? Nutrient reductions have led to SAV recovery since the 1980s, and how are they related to the human activities? The nutrient pollution reduces SAV, and if you ameliorate that pollution, you're going to get resurgence. And the species enrichment, if we get multiple species, that enhances the SAV. So that's the third paper that's, like I said, part almost out. And then our fourth and final analysis was the segment analysis. Now, this wasn't easy. There are 97 segments around the bay, and to try to take water quality stations that are not located in the right location of SAV, and then we got this issue that the conditions inside are different than outside. The temporal integration of water quality versus the SAV is an issue. We have events like the heat events we've talked about, but also storms like Tropical Storm Lee. We have interannual variability versus long-term trends. We have local effects, shorelines, seed banks, land use influences. So it's not easy. Let's put it that way. So it didn't come out to be a simple story, but we came up with a way around that. And I'll go through that. So here are the three examples. What we did is we took the 97 segments, we combined a bunch of them, and you'll see some examples of combined, like there are two segments here that we combined and several here. And I'll go through three examples: the Flats, the Upper Potomac, and the York River polyhaline. Here's what we're going to produce for each of those segment analysis. A front page, a first page, and this is going to be web-based mostly. And this will have the big story, the executive summary, the trends, and the conceptualization and the take-home points. And then we back it up with much more detailed information, including references and web links. So going through the first page, here's the Flats. We have an executive summary in simple language, talking about the recovery that occurred and the fact that Tropical Storm Lee didn't wipe it out completely. And then we have a graphic that we've created, which is not just the area, which is the top bar, and that's the stuff you've seen for years in the SAV data, but now we have the density layered into that. And so from that, you really see that resurgence isn't just an area, it isn't just the doubling of area, but the density of those massive beds up there. We see the Tropical Storm Lee effect, but we're seeing the recovery from that there. And then we put that into a pictogram here that shows the thin, wispy bed prior to the 90s, the resurgence, the large, dense, multi-species bed here, and then the resuspension, the Tropical Storm Lee and resuspension effects. And now we've got an issue associated with expansion of a cyanobacteria, Lyngbia, that's occurring there. And then these take-home points we've spelled out, and then we have bulleted on the front page, but then in the back we explain and provide links and references to each of these. So first of all, on a management context, clearly the goal is attainable, and that's fairly easily seen because we attained the goal in right before Tropical Storm Lee. So it's certainly attainable. It's also susceptible to extreme runoff, but resilient. You have to worry about resuspension, changing patterns in the diversity and habitat quality have occurred. We have this expansion of the cyanobacteria lyngbia. There's some management implications we listed and some references. So that's the format, and that's how we did the Upper Potomac. You can see the same style. I'll go through those quickly. We have our executive summary. We have our density and abundance graphic. We have our conceptualization with the Blue Plains upgrade, and then the resurgence of particularly hydrilla, and then the native species coming back in. And we have our footnotes with the attainability and the hydrilla and all those points made here. And similarly with the York River, we went through and generated a summary, a density graphic, a conceptual diagram and timeline with footnotes if we can follow that trend. For all the rest of these throughout the bay, we'll have a complete set of these segment analysis that will give managers sort of a working tool to work with. So kind of looking back on our SAV-SYN effort, we produced 3 papers working on a segment analysis. 2 of the papers are published, one's close, it's in review, and we're looking as a end date for the segment analysis April of next year. We also posted some blogs about the workshop on the Integration Application Network website. We even wrote a little poem about the Chesapeake Sentinels. We also reflected on, as a group, what worked in this synthesis effort. So some of the lessons learned were that you needed people who've done this. Bob Orth and I have done this before at the National Center for Ecological Analysis. You need people who've done this. Limit the size. You have multiple immersive workshops, regular communication, flexibility, adaptability, product focus. We knew we wanted the segment analysis in the papers. Conducive location, we used Annapolis and Solomons as places where we could go and have this immersive activity. Clear goals and objectives. We had a lot of fun along the way. Next time we recommend including graduate students and staffers, having slightly longer immersive, maybe 3-day instead of 2-day workshops, and a little bit of flexibility on the project funding so we can do some analysis. Just a couple weeks ago, we presented these results. Cassie Gerbitz and Chris Patrick made fantastic presentations at the biannual Coastal Estuarine Research Federation conference in Providence, Rhode Island. And here's our group in living color and our little coffee mug that demonstrates that a little caffeine goes a long way when you're doing synthesis. So thanks very much. I hope I was able to get across the essential findings of this synthesis and encourage the Bay program to move forward towards more of these kind of synthesis efforts. They're very productive and very useful and very practical and management oriented. Thanks.